How to change my background.
Okay, Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon, ladies and gentlemen. Welcome to the webinar titled Biology and COVID-19 Mitigation, organized by the Faculty of Science and Technology, Universitas Medan Area. On this occasion, I would like to thank to the committee appointing me as a moderator, so that I have an opportunity to meet with special figures and various people with different background, professions, and place of origin. The Honorable Director of Universitas Medan Area, Professor Dr. Dadan Ramdan, MSc, MN, the Honorable all speakers and the participants attending the webinar. Previously, I would like to greet the speakers who will deliver the presentation today. Good afternoon, Dr. Nilanjan Deb.
Good afternoon.
How are you, Doctor?
I'm very fine now.
I hope you will enjoy within the next 3 hours webinar.
Sure.
Okay. Good afternoon, Mr. Muhammad Yani bin Abu Sama, PhD.
Yes. Good afternoon, everybody.
Nice to meet you, although we are not meet really physically.
Okay.
I would also like to say hello to Professor Diana Waturangi. Good afternoon, Prof.
Ya, Pak. Sore.
Good afternoon, Prof.
Ya.
So we met again today. We've discussion a couple of days ago. From last conversation, you know that Prof. Diana was graduated from the same university.
Right.
That's IPB University.
Yeah.
And the last but not least, I want to greet Dr. Faisal Amri Tanjung. So he is a Dean of Faculty Science and Technology of University Medan Area. Good afternoon, Dr. Faisal. Not joined yet. Today we'll talk about a very up-to-date topic that is related to our life, which has taken up most of our time, which may have haunted our minds. That is coronavirus, namely COVID-19. Soon, we listen to the speakers, we listen to the exposure of experts and experienced people, how biology could mitigate the spread of COVID-19. However, previously, we need to listen to the opening speech of Rector of University of Medan Area, and then we'll ask our Rector will open the webinar officially. Thank you to Rector for coming, and now time and place is yours. Please, Prof. Dadan Ramdan.
Okay, thank you, Prof. Jo. Bismillahirrahmanirrahim. Assalamualaikum warahmatullahi wabarakatuh, and good afternoon. The Honorable Professor Dr. Nilanjan Deb from the Institute of Agriculture Science, University of Calcutta. The Honorable Professor Dr. Diana Waturangi, MSc, from the Faculty of Biotechnology, Atmajaya Catholic University. And then the Honorable Muhammad Yani bin Abu Samah, Master of Engineering, PhD MBA from the Fortis Institute, Scranton, Pennsylvania, USA. And the Honorable Dr. Faisal Amri Tanjung, STMT, Dean of Faculty of Science and Technology, Universitas Medan Area, and also the Honorable Professor Dr. Insinyur Zulkarnain Lubis, MS, Head of Doctoral Program of Agricultural Science, Universitas Medan Area. And there are participants of Faculty of Science and Technology webinar entitled Biology and COVID-19 Mitigation. First, we want to praise and thank to Allah Subhanahu wa Ta'ala, who has given His blessing to all of us, being grateful with healthy and prosperous, so we can able to attend the seminar today. Ladies and gentlemen, as we all know together, COVID-19 pandemic has been a serious globally issue across the world due to its great impact on various fields, especially in social life and economic growth. COVID-19 pandemic has involved various academic fields to help overcome this condition, including biology. World Health Organization, WHO, has stated coronavirus disease, COVID-19, is an infectious disease caused by a newly discovered coronavirus. Biology has closely related to the outbreak, since virus or virology is one of branches in biology. Ladies and gentlemen, the close relation between biology and COVID-19 pandemic is one of the basic reasons to raise the topic biology and COVID-19 mitigation in this event. As we know, various mitigation strategies of COVID-19 have been done to reduce the transmission of the virus. Many studies related to the virus have been done by researchers, including biological scientists, such as researchers about vaccine, medicine, health equipment, and the virus itself. Collaboration between researchers across the countries is very important to accelerate the achievement of results. We have great expectation that all of this effort will help us to deal with virus as soon as possible. Ladies and gentlemen, this seminar can be one of the media for us to discover the role of biology in COVID-19 mitigation. This prevention efforts has been done by government and community along this time, such as the implementation of health protocol in every activities during the pandemic, and the policy of new normal as socialized by governments. The issue of finding the right solution to deal with this pandemic is still developing by the scientists around the world. Therefore, it will be an interesting topic to talk about this COVID-19 pandemic through biology point of view, especially the mitigation efforts to reduce transmission of the virus. The material will be delivered by great speakers, and I hope it will explore our knowledge and awareness towards this pandemic. Finally, I hope the seminar will give good contribution and positive impact toward the effort of overcoming this COVID-19 pandemic. Thank you for all the speakers and committee for all the hard work to make this seminar can be held today, and also for all the participants. May you and your family are always surrounded with prosperity, health, peace, and full blessing from the Almighty God, Allah Subhanahu wa Ta'ala. So, in the name of Allah, Bismillahirrahmanirrahim, I open the seminar officially. have a great seminar, and thank you for your attention. Wabillahi taufiq wal hidayah. Wassalamualaikum warahmatullahi wabarakatuh, and good afternoon.
Thank you, Prof. Dadan, for the opening speech and opening the webinar officially. And now we'll begin the presentation by the speakers. Every speaker will be having maximum 25 minutes for presentation. So we have spent an hour and 15 minutes, and then we'll continue to the Q&A session. Questions should be submitted in writing via the available chat facilities. And then after the presentation, the question posted by the participants will be read out by the moderators, and the speaker will be asked to answer the appropriate question. If there will be plenty of questions, only a part of them that will be read and answered subject to the time available. So while the speakers presenting their papers, the participants are expected to pose their question so that the moderator has time to compile and select the questions. Okay, now let's get to the presentation. And I'd like to invite Professor Dr. Diana E. Waturangi to be the first presenters. And before that, let me first read her CV. Wait a moment. Okay. Professor Dr. Diana Waturangi right now is the work for the Faculty of Biotechnology at Majaya Catholic University. I think I read all of the CV. I just read the education. Professor Diana Waturangi graduated from Bogor Agriculture University for her bachelor, and also for master the same university, and also the PhD. So all the degrees that PhD, master, and bachelor of Prof. Diana got from Bogor Agriculture University. So she has a lot of publications. I will not read it one by one, so you can see it from my posting here. He has also a lot of academic presentation that has been done, and a lot of research grant and awarded. She has an editor of some journals and some general lectures in various universities and workshops. So I think he is very expert, very experienced in his field. So now I think I would like to ask Diana to present his paper. So Prof. Diana, time and place is yours.
Okay, thank you, Prof. Dr. Zulkarnain. First of all, I would like to thank the organizing committee and also especially to the rector of University of Medan area. Thank you very much for inviting me to present the topic for today. It's about COVID-19 and possibility of secondary infection, and also alternative way how to overcome the antibiotic resistance that usually related with bacteria. As we know that coronavirus disease is RNA polymerase. They have RNA as a molecular base, and they also have spike protein while this protein recognize the ACA2 receptor in human cell, then this receptor we can find in nasal and also in mouth and also in the eye of human body. In this slide, I would like to show about the size so that we can make comparison the size of the flu virus. Of course, this is bigger than molecule like nucleic acid, like protein. So the size is less than 100 nanomolar. Nanometer, and also, but they are smaller than mitochondria and also bacteria in general. That's why we cannot use the fabric regular mask to protect us from the virus. The coronavirus disease is highly transmittable, and this is pathogenic viral infection due to caused by we call it SARS-CoV-2, and it emerged from Wuhan, China, and then spread around the world. The classification is they are grouping of nidovirales, and the coronavirinae. There are 4 groups of these coronavirinae, which are alpha, beta, gamma, and delta. But the one who can infect mammalia, including human, is alpha and beta. When we make comparison between SARS and MERS and also COVID-19, the COVID-19 coronavirus disease 19, they have same receptor with SARS, which is ACA2, but different with MERS. And epidemiological comparison, we can see that the incubation time different between flu virus and SARS and also MERS. And also the case fatality rates also different. Actually, it's not that high that SARS, but we have to consider that this data is still ongoing. So the transmission of the coronavirus, SARS coronavirus 2, it's actually phylogenetically and genomically, this is similar with SARS-like bat coronavirus, but the intermediate after bat virus and then to human, actually it's still missing link. So this is still question mark, the intermediate host. After that, we can see that it starts human-to-human transmission. That's why it is important to keep the safety protocol as physical distancing. So actually, we can see that this virus revealed that there is a homologous recombination between clade A, which is bat coronavirus, and the origin unknown isolate. And the location of the combination is in the spike of the glycoprotein, which is play a role to recognize the cell surface of the receptor. And this virus also has a more similar genetic information with bat coronavirus, but they show also similar codon usage with a virus that infect snake. So the recombination of the SARS and in the spike glycoprotein, this is people predict that this is play a role as initial cross-species transmission from bat to other mammal. So now we look at the life cycle of the coronavirus. The coronavirus recognize the ACE2 receptor in human cell. So they start to insert the cell and then fusion and uncoating and then start to express the replication, transcription, and translation. And after that, they do the multiplication of the cell, of the virus cell, and then do exocytosis and start to find other cell to be infected again. So this is all the life cycle of the coronavirus. So actually, what happened inside the body? In general, there are 4 phases of the start from cell invasion until the acute respiratory distress syndrome. The first phase, we looked at virus start to recognize the ACE2. They can enter. So the entry point is from nasal cavity and also the mouth, the tongue, and as well as from eyes. Because these 3 parts is we can find the ACE2 receptor, which is can be recognized by the virus. So this all the 3 points, and after that they go through the lung, and in the lung the ACE2 receptor located in the top of the lung called pneumocyte. And in the German viral study, it showed that 50% of the patient that on day 7, they show IgM and IgG antibody. And after that, the phase 3, several people can getting more worse because after that they can have pneumonia. And phase 4, it can be happen also with several people acute respiratory distress syndrome. And the most important thing is cytokine storm. As we know, cytokine is very important as for human immunity, which is cell defense. How to when we get infection, and then our body start to produce cytokine to make cell die. But in the case of COVID, the production of cytokine is 50 times higher compared with the infection in Zika and Nile virus. So we can imagine 50 times higher, and it make the cell die become spread all the body, and also can cause organ failure. So this is the view of the healthy lung and also infected lung. When we look the infected lung, there is increasing of mucus secretion and also the thickened muscle. So people usually difficult to breathe. So what about the secondary infection? Because as we know, sometimes when people got infected with virus and then the immunity suppressed, and usually it is easier to get infection, which is we usually call co-infection. Many hospitalized victims are developing also potentially lethal secondary co-infection, such as bacterial pneumonia. And COVID-19 patients need to be tested not only for coronavirus only, but also to detect the bacteria, infection with bacteria and fungi. Because for some people, it is very lethal also. So it is important also to diagnose not only the virus, but also the bacteria. In the case of respiratory viral infection, the infection may increase disease severity and mortality. And in 1918, when influenza outbreak happened, it's also in SARS and MERS. Also, we can see that it's also usually related with bacterial infection, so not only virus. And data regarding superinfection in COVID-19 are still limited and emerging. So diagnosing co-infection is complex because sometimes people already have the infection as chronic infection. So actually, most of the patient already have the infection of bacteria before, so made the case become more severe. So far, these are the list of the bacteria from the several publications that show play a role as co-infection like Mycoplasma pneumonia, Legionella, Acinetobacter, Klebsiella, and also Streptococcus pneumonia. So usually we know that to treat the bacterial infection, it is important to use antibiotic. But we have to remember that antibiotic do not treat the virus, such as COVID-19 as well. But they're absolutely essential for treating the bacterial infection. But the problem is we found that usually pathogenic bacteria is common resistant with antibiotic. So there are two types of antibiotic resistance actually. Firstly is natural intrinsic, where the bacteria already have the gene that can produce something and then to inhibit or to destroy the antibiotic. And the second is we know that usually the pathogenic bacteria also form biofilm. So this is different resistant property because with the producing of biofilm, bacteria have protection like physical protection. So therefore the antibiotic cannot cannot insert, cannot attack the cell because they already have the physical protection, which is usually exopolysaccharide. So this is the biofilm formation in the lung, where bacteria start to colonize and then develop biofilm, and also the microbial proliferation lead to acute pulmonary exacerbation. So this is very important. We need also to overcome this problem, especially the antibiotic resistance related with producing biofilm. So this is important to find alternative way how to treat the resistance bacteria, usually the one who develop biofilm. So it is important also to develop antibiofilm as alternative way to combat antibiotic resistance. First is we know before biofilm formation was formed by bacteria, the bacteria need to communicate each other. So for each species of bacteria, they communicate and then start to form the biofilm. So it is important that we explore compound from other bacteria that have capability to inhibit the communication so that the biofilm cannot be formed. And the second approach is to find also from other bacteria, because we know the lifestyle of the bacteria is strong with competition. So we just need to find the exploration of which bacteria have capability to inhibit or to attack or to destruct the biofilm structure of pathogenic bacteria. So actually there are two approaches of this. As we know, this is the antibiofilm application is we can use as a preventive, usually in the medical device, can put the antibiofilm as preventive action, and also to use it as a treatment combined with antibiotic, so that we can find the antibiofilm who can destruct the biofilm formation, so that after that, together with antibiotic, the antibiotic can be used to attack the pathogenic bacteria. So this is our study so far that we found from many bacteria from the phyllosphere and also from marine bacteria, which have the capability to inhibit or to destruct the biofilm formation from pathogenic bacteria. These are some of the literature. I think thank you very much for listening. Overall, this is we need to think also not only of care about the coronavirus, but we have also to consider the secondary infection, which is caused mainly by bacteria. And the bacteria, it is important to treat with antibiotic. But since a lot of bacteria resistant with antibiotic that normally caused by biofilm. So it is important also to explore and to find the antibiofilm property, which is can help to treatment the secondary infection. Thank you very much.
Thank you, Prof. Diana, for the presentation. For the participants that are going to give a question to Prof. Diana, just keep your question first, or you can set by chat, and then Prof. Diana will answer the question after this presentation session. Okay, now let's go to the next speaker. I think Dr. Faisal Amri Tanjung have already for the presentation, but before that, I'd like to read first the CV of Dr. Muhammad Faisal. Dr. Faisal Amri Tanjung. Okay. Faisal Amri Tanjung now is a senior lecturer and the dean of the Faculty of Science and Technology, Universitas Medan Area. He earned his PhD in Polymer Engineering distinction from the University Malaysia Perlis. And prior to join with UMA, he worked as a material scientist at the Alcon Siba Vision Sendirian Berhad and a lecturer at the Department of Chemical Engineering, Cochin University, Malaysia. His current research interests include bio-nanocomposites with emphasis on structure, property, correlations, interfaces, and polymer functionalization. Okay, I think Dr. Faisal, now is your turn. Go ahead.
Okay, thank you, Professor Jun. Can you hear me?
Yes, of course.
Okay, thank you. Very nice introduction. Thank you for the opportunity. Assalamualaikum warahmatullahi wabarakatuh, and very good afternoon to everyone who's watching this international webinar, which is organized by Faculty of Science and Technology, Universitas Medan Area. Okay. Screen. Okay. 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 Can you see my presentation? Okay. Yes.
Okay. Okay, just continue.
Today I will. Today I will deliver my presentation is about graphene and coronavirus. Maybe some of you don't know me. Let me introduce myself. My name is Faisal Amri Tanjung. I'm currently a dean at the Faculty of Science and Technology and also a university associate for the Carleton University. So let's just start the presentation. Okay, maybe some of you already know what is the graphene. Graphene is a material made from carbon. This is one of the new class of the material that is only one atom thick. So that's why so-called two-dimensional material because they just have two-dimension length and width. And because of the thick is only one atom is considered zero, so it's negligible.
Dr. Faisal, I cannot see your presentation, I think. Could you please just share?
You cannot see it?
Yeah. Just share the slide.
Okay, sorry. Okay. 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 Can you see?
Yeah,
Okay.
can see now.
Here you can see now. Okay. This presentation entitled about the graphene and coronavirus. Okay. Let me start from uh, the first. Okay. Okay. Okay, what is graphene? Graphene is it's a material made from carbon. You know that these are the many actually the material made from carbon like fullerene and graphite. But graphene is one they consider the new class of the carbon-based material. It's invented in 2004 in from the by the a group of researchers in the Manchester University. It represents a conceptually new class of the material that are only one atom thick. So that's why they call as a two-dimensional material because they just have a two dimension, is a length and width. Since the atom is the thick is only one atom is considered negligible, so it's considered zero. So that's why usually they 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 consider this material as a two-dimension, and then it has many different properties which make it potentially useful. I think we can discuss this later. What is the factor that make this material is a very useful according to the different properties, and it's expected to replace common material like plastics and silicon on certain products only as the next decade, because some of the product derived from graphene can be used as the stretchable and ductile material that can be used as the sensor. So it's a very um, what is called is like a deformability. This is if you look at the picture, this is the picture that shows the hexagonal area of the graphene itself, and is and the below one is like in the powder form. So in a nanometer scale, this graphene is actually as composed of the layer by layer surface carbon. So that's why in some of the even the researcher from in the material, they call that is like a sequence AB sequence, which is between one layer and one layer by layer stacking in the AB sequence. So this is a graphene sheet are building blocks for other graphitic material. We can see that many forms that can be obtained from this material. For example, like we can see that this is a stack on the top of each other that make graphite. We see that 1 millimeter thick graphite contains about 3 million layers of graphene. So that's why this is called that the graphene composed of the layer by layer of carbon. It can be also a roll up so that they make a carbon nanotube. You know that the carbon nanotube is very promising material now used for catalyst, even the photocatalysis, and then also for bioremediation, for environmental application also. It also can be cut and folded into a spherical shape to make the like a fullerene. Fullerene, you know that is C60. C60 usually can be used as the hydrogen gas storage material. So we move on to the properties of graphene. Graphene is first is the great semiconductor because the electron can move with minimal resistance. So this is very good material in the electronic application. The second is extraordinarily strong because it's 100 times stronger than steel. And so that's why some of the graphene also is applied for the structural application, like for automotive application. We know that like Tesla, even now they produce car that the exterior design is composed from graphene, no longer use like a steel and other composite material. The third is the thermal conductor, because heat can move 10 times faster in graphene than in copper. So it's a very good conductive material. That's why in many applications, graphene also is used as a solar cell application. The fourth one is a nearly transparent, because 97.3% transparent due to its only being one atom thick, nearly transparent. So that's why this kind of the properties is exploited to be used as the sensing devices. Another one is the flexible and malleable, because they attributed to its lightweight and thinness, because it can flex 20% without damage. You can see that the figure, it's how the material can be flexed almost 20% without damage. And potentially to be cheap, affordable, because carbon is incredibly abundant on Earth. Currently, graphene is obtained from the biomass waste. We know that biomass waste, it can be banana, coconut trunk, many things like tree, and then it can be carbonized into the carbon, and then from the carbon it go to which process we call that graphitization to get the carbon with high crystallinity and order structure. And then another one is incredibly thin, because only one atom thick. So that's why it's called a two-dimensional, because only one atom thick is considered negligible. This is the graphene extraordinary properties. We already discussed earlier, but we can see that this is key graphene properties. First of all, is the atomic thickness. This single layer is only one atom thick. So that's why this material called two-dimension is about 0.335 nanometer. And how about the electromobility? We see that the highest electromobility of all electronic material theoretically of 200,000 is almost 100 times higher than silicon. So this is a very good material to replace silicon. Strength, defect-free. This is a monolayer graphene. Is the strongest material ever tested with a strength of 42 Newton per meter, which equates to an intrinsic strength of 130 GPa. It's even 100 times stronger than the strongest steel. So that's why many construction material and for automotive and for plane now use this kind of material, because this is strength. And then also lighter compared with the other mineral, compared with the other mineral materials. And also toughness and stretchability. Although graphene is relatively brittle, but it can be stretched by up to 25%, highly relevant for flexible electronic. This kind of properties also is being exploited as the material for the sensing devices. Stiffness. Experiment on defect-free graphene monolayer have yielded a young modulus is 1 terapascal. I think it's very high. One of the highest value of any metal, about the same as diamond. And impermeability. Impermeability, we can see that impermeability. We see that even the smallest atom cannot pass through a sheet of graphene, because it's a package structure of this kind of material. So it's a very good to be used as what we call that is like anion exchange membrane, for the energy application, energy harvesting application. And because high surface area is a 2,630 meters square per gram, with less than 3 grams, you could cover an entire social field. And then electrical resistivity. This is among we see that among the lowest of any non-material at room temperature, even less than copper. And also thermal conductivity is which is due to the structure of the metallic bonding between the carbon structure. transparency is absorb only 2.3% of reflecting light. So that's why it's a very a good promising material to be used as the sensor. Now we move on to the application of graphene. This is the some of the key application area for graphene, like for electronic, for membrane, for sensor, for composite and coating, energy harvesting and storage, and also biomedical technologies. According to the topic, this webinar, we take the topic about the biological application of this kind of material. So we focus our application into the biomedical technologies. If we can see the biomedical technologies, graphene is paving the way for novel biomedical technologies because thanks to high surface area, electron mobility, and functionalization potential. Because the graphene bioelectronic has become a groundbreaking field that offers exciting opportunities for developing new kind of biosensor capable of establishing outstanding interfaces with soft tissue. So this is one of the explanation why graphene is very considered as the promising material for biomedical technologies. Some of the example of the application that has been explored currently is the like for thermal ablation for highly resistant cancer cells, targeting and neutralizing cancer stem cells, bioelectronic bioassay, electronic interface to living cells and nerve tissue, and luminescent graphene text with for bioimaging. So we can see that the graphene is considered as the multifunctional material because many things that we can pull out from this new class material. Now, based on the current knowledge on hazard of nanomaterial, a number of characteristics can be identified that may be relevant to hazard assessment of graphene. I think it's a very important aspect that should be considered if we have a willingness to use this kind of material into the medical application. First thing that we need to consider is the thickness, because the biological response to 2D dynamics is unique, and the physical interaction of graphene cells are expected to vary based on the aspect ratio and mechanical properties. The characterization of lateral size and thickness of 3D material is critical. This is considered as the intrinsic properties of this material that give a specific surface, not a specific response to a particular biomolecule that acts on this surface. The second is the composition. Chemical composition is one of the most important considerations for determining the biological interaction and fate of the graphene in vivo. Surface chemistry and dissolution of the matter will be determined by it, which will be in turn affect the cellular interaction, uptake, and biodiversity. Sorry, biodistribution. And this is very consideration also that need to study more the effect of composition. And the chemical transformation. As a layered material are chemically exfoliated, the fundamental properties of the material change. Similarly, biological interaction with layered material vary as a function of exfoliation state. We know that since this material composed of layer by layer carbon, so it can be intercalated or can be exfoliated. So many researchers try to measure the degree of intercalation because the degree of intercalation will determine the ability of this material to react with a certain molecule. And in order to get the graphene into the nanoscale size, usually they will try to synthesize graphene oxide, which is has a smaller size than graphite, because we see that at first Graphene is called a graphene platelet. Usually, in the native in pristine form, it's called a graphene platelet. After that, we exfoliate it using strong acid or maybe strong base in order to get the exfoliation state. So that's why the graphene will change into the graphene oxide, so we can get the material into the nanoscale size. So this is when we use this size, then we can use this as like material to detect the kind of the biomolecule, because we see that for example, like now COVID-19 viruses is a size is around 100 to 150 nanometer. So means that we need the material which has a larger surface area and also a smaller size than the virus itself. Because if the material uh, the size is higher than the virus, it not possibly can detect the virus. So it's a very important to enhance the efficiency of this material in order to recognize a particular biomolecule. And also the surface functionalization. Generally, the surface functionalization of graphene will dictate the dispersion stability, the surface reactivity, and the degradation behavior of which will contribute to the overall toxicity of the material. So this is the common issue now, how we get to functionalize the surface of this material in order to enhance its reactivity, biocompatibility, and solubility into the particular material which attach on its surface. So the surface functionalization is very critical issue currently. Now, many researchers they try to functionalize the surface of a graphene using various biomolecule in order to enhance its efficiency. And also the structural form. Structural form is relate to what kind of the application when you use the graphene. Usually, when you use if you want to use as a sensing device, you need to reduce the size into the nanoscale. Means that you need to exfoliate this material. If you want to use this material for the structural application, for automotive application, you need to make it as a layer, as what we call is a thin film, but very high toughness thin film. So it's a depend on the what kind of application you want to build up from this kind of material. Okay, now we move on to our main topic today is about the graphene against coronavirus. We see that this is the graphene. If you look at this figure, this is a graphene structural program. It's a hexagonal array of carbon, and this is the coronavirus. We can see that the protein spike on the surface of the virus membrane. We see that we know that the coronavirus is the unprecedented phenomenon outbreak of the coronavirus disease 2019. So what is called as a COVID-19 is tearing global health system apart. The coronavirus is formed by surface projection on the viral lipid envelope enclosing single-stranded positive sense RNA. I think Professor Diana already explained what is a coronavirus itself from the molecular biomolecular point of view. It's very good explanation actually. And why many researchers are interested to use a graphene for to combating to tackling the effect of COVID-19 pandemic. So this is one reason why graphene has become get much attention to be used as the one of the candidate material for the COVID-19 mitigation. Graphene has captured much attention due to its superb electronic properties, including approaches to fight or detect drug-resistant bacterial infection. Yeah. many report already published about the advantage using graphene to tackle the bacterial infection. So that's why many researchers now try to explore this this potential of this material. Is it is it will no, is this kind of material can be used to combating the coronavirus? That's still a question of remark. Now we go that the next presentation. The next slide is about the some strategy how we enhance the reactivity and the biocompatibility of this material like a graphene. Now we consider about the graphene and graphene oxide like using the biofunctionalization is one of the strategy usually used by the researcher to enhance the biocompatibility, solubility, and selectivity. We know that we look at the figure A, we see that this is an epitome of graphite form that we can be transformed into the various form like layer by layer, tube, and also like a spherical form. And if you look at the figure B, this is the schematic image, for example, representing a loading of doxorubicin and camptothecin onto fatty acid modified graphene oxide. This is why one of the studies that have reported in the literature using the fatty acid and the camptothecin and doxorubicin to enhance the surface activity of the graphene. The aim of this biofunctionalization is to improving their biocompatibility, solubility, and selectivity. Now this is the summary of some of the literature reported the functionalization of graphene using the various biomolecule. If you look at this, graphene and its derivative have been reported to be functionalized with avidin biotin, peptides, NAs, protein, aptamer, small molecule, bacteria, and cells through physical absorption or chemical conjugation. And then functional graphene biosystem with the unique properties have been used to build up biological platform, biosensor, and biodevice. So many biomolecules have been used to functionally to react your functionality to modify the surface of the graphene in order to get the graphene with desired properties for the particular application. Now we move on to the graphene material interact with viruses. So the first study about the potential of graphene to combating the virus began in 2012. In this study, a thin film of reduced graphene tungsten oxide has been exploited for the photo inactivation of bacteriophage under visible light irradiation. So this is the what we call maybe as the prime mover in the research in using graphene oxide and reduced graphene oxide to combating the viruses. And large surface of graphene provide the highest ligand contact area for the absorption of negatively charged, which can interact with virions, positively charged residue, and block microorganisms. This is one of the reasons why graphene has been used as the material that can be combating the virus, because it has firstly it has a large surface area, right? Because it's a specific surface area can contact directly with the molecule surface. And the second, because the charge of the surface of the graphene, we know that Before the modification of the surface graphene is actually is a neutral charge, and while the ferrion surface is a positive charge. In order to increase the selectivity, in order to reactivity of the graphene toward the ferrion surface, we need to change the neutral charge on the surface of graphene into the negative charge. And one the strategy is is using functionalization strategy. We need to choose a kind of the chemical or biomolecule that have a strong charge, negative charge that can attract the ferrion surface, because the positive and negative when come in contact, they will attract to each other. Reduced graphene oxide sulfate, for example, demonstrated their antiviral activity against African swine fever virus, orthopox virus, and herpes virus strain. So this is the previous report or studies that already published, and can give get some insight. Is it the potential of this graphene for the mitigation of the COVID-19? And also the using of beta-cyclodextrin, which is a functionalized on the surface of sulfonated graphene to treat the respiratory syncytial virus. And also the use of polyglycerol sulfated graphene functionalized with fatty amine, chain warp, and inactivate SAV1. If you look at the figure A here, this is the main structure of coronavirus. We see that there is a membrane protein, RNA, nucleocapsid protein, spike protein, and envelope protein. And see the figure B, how the functionalized graphene break down the surface of the ferrion membrane. See that how the representation of SCV warping by sulfated graphene derivative and illustration of long alkyl chain disrupting virus envelope. So this is the how the mechanism to inhibit the spread of the virus in order to protect the living cell from this bacteria and also the virus that can damage the living cell. Now we move on to next slide. This is another application of the use of graphene to tackling the COVID-19 pandemic. For example, graphene textile for pandemic spread control. Usually, we see that one of the key areas for the graphene application as the membrane, and as membrane is same like as a nanofilter. Graphene filter has been produced for capturing particulate and bacteria to decrease the transmission of nosocomial infection, and also the bacteria blocked on the filter are not able to proliferate, and by increasing filter temperature over 300 degrees in microorganism along the molecule that can cause disease are destroyed. This is one application maybe that we can build up in hospital, maybe in office, so that you can protect the environment from the spread of the virus. We can see that in the figure how the graphene textile has been used to protect or maybe to tackling the pandemic of the virus. We see that the graphene has been modified, has been transformed into the sensor. You see that the sensor arrays implemented on textile and interface with flexible electronics and can report the location of positive and identify source of the outbreak. I think in some developed country, they already established this technology where someone can get the information which area is safe or and which area is not safe. So they can identify which area is categorized as a red zone and which area is categorized as a green zone by using this technology. And another one is we look at the figure below is the use of the graphene as the face mask. The face mask is made from graphene has a advantage as the what we call is uh, very good protective barrier as compared with the conventional face mask, because it could be recycled by photocatalysis or heat. So it is can be after use, the face mask can be simply heat up using a sun ray or in just live on the sun ray is around 2 to 3 hours before the person can reuse this fast. Now we move on to next slide. This slide is one of the study from a group of research and from China actually. I take this research for you to give the example how the functional as a graphene oxide using the herb like a curcumin. I think you already know what is curcumin. Curcumin is a natural polyphenol which is isolated from the rhizomes of the perennial herb Curcuma longa and holds various functions including antioxidant, antifungal, antibacterial, anti-inflammatory, and antiviral. This polyphenolic compound has gained significant attention due to a variety of biological activities and low toxicity. So that's why, especially in Indonesia, curcumin has been used as a traditional medicine for person who got the flu or maybe a cough. And we see that this figure A, for example, this is how the synthesis of functional nanomaterial composite. We see that there is a graphene and graphene, and then it's filled with CD or cyclodextrin, and then and then the curcumin biomolecule attached on the surface of the graphene molecule. So when two or three material combine into each other, so we can consider it as a composite, because its composite will have a different properties compared with the parent material itself. We see that the figure B, how the proposed inhibition mode of the functional nanomaterial composite against RSV infection. We see that how the composite material of the graphene inhibit the virus molecule approach the living cell. So some of the virus are also broke by the composite material. We see that the introduction, the explanation here, that the introduction of the sulfonate group to the functional graphene surface can mimic the cell surface and inhibit the RSV infection through a competitive inhibition mechanism. Yeah,
Excuse
maybe
me,
some.
would you please make it shortly, because the time is about to.
Okay. Just two slides left. Okay. And the synergistic antiviral activity is due to the nanomaterial themselves and drug curcumin. So some of the material, the biomolecule has been used to functionalize the surface is not only to enhance the activity but also to increase the binding what we call the binding traction, and also to act as a biomimicking surface, as also as a biomarker. So that can increase the selectivity of what kind of this material into the particular biomolecule. Now we move on to the end of the presentation. There's a limitation and challenge when someone try to use graphene as the material for combating the coronavirus. First, the immediate use of graphene for treatment of COVID-19 is unrealistic for the current moment. The only available in vivo test for virus treatment demonstrated the efficacy of GO hyperizin in darkly infected with the novel dark virus. So it's never been reported the use of graphene as a drug for human. So there is no experiment using graphene as the drug that the person can intake as into their body, because yeah, we need to consider the toxicity of this material. The only available data about this using of this material is only for duckling, which is infected with novel duck virus. So the in vivo toxicity of graphene is still a matter of debate, and graphene toxicity are made difficult due to the infinite combination of dose, surface chemistry, exposure ratios for the evaluation, and also because its instability and aggregation in solution are other problems, and given the mandatory stability required for drugs and vaccine storage prior use. And this is some reference that I use for this presentation. Maybe some of you, if you're interested in this kind of this topic, maybe you can refer to these journals. And thank you. And back to the moderator. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Faisal, for your presentation. And now we're going to be the next session. The speakers are Dr. Nilanjan Deb and Muhammad Yani bin Abu Sama PhD. I think I will ask first to Dr. Nilanjan. Dr. Nilanjan, but before that, I would like to read a little bit his biography.
Yeah, hello. Good afternoon, everybody. And my friend Yani is there sitting. Yeah.
Yeah, okay. This is the CP of Dr. Nilanjan. He is an associate professor from University of Calcutta. He has a lot of research and the, papers. He also is interested in biology, agriculture. But I think I will not read all in detail all these CV, and I'd like to ask Dr. Nilanjan to present his papers today for this webinar. And now, Doctor, time and place is yours. Please.
Yeah, thank you very much for showing this thing very, very most humbly. I put my all respect to the who has invited me, the rector of this Medan Area University. Good afternoon, all other participants there. And I will take few minutes to talk about this thing, because now the time is to work rather than to speak. there are areas where I have done that biography was there, maybe more than 12 or 13 areas, electronics to biology. And I had more than 4 or 5 dozens of US patents which were purchased by the US companies. Now here I am directly coming to the point of COVID, because this is a pandemic here for last 3 months. It is havocing throughout the world. Now today the statistics is 4.7. I think that more than that, 0.47 million. That is 4.7 lakh people are affected, and it was diet and affected is it is going to be very, very high, staggeringly high. And in our countries, in Asia also, we are suffering, we are going to suffer in future. And there is no, neither vaccine nor medicine nor any curative thing so far we have found. But here I will directly go into our mitigation proposal, and I will give you that information. Already I have dispatched the formulations to my friend there, Muhammad Yani. He will be connecting with you with the samples, and I would like that has to be validated as early as possible on the COVID-19, that is SARS-CoV-2 directly. And till date, the information I am sharing with you, these formulations are really working, and my preliminary studies and testing has proved that these formulation has the potentiality to control the spread of these highly contagious, which has become a pandemic, SARS-CoV-2 or COVID-19, among the population who are going to be breaking the lockdown, who are going to be the infected areas, who are going to out for the job. Now, let me come. There will be questions how this medicine will work and why this is not there. First point is that this is a formulation which is made out of edible ingredients, and the mechanism here works just similar to that of hand soap mechanism. Hand soap, when you wash when the WHO and UNESCO and many other agencies across the globe have suggested, and it has proved that the viruses and the bacteria which has lipid layer on their bodies, on the outer surface of their protein layer, that can be disintegrated, and the virus gets immediately killed within 20 seconds if it comes in good contact with the soaps or emulsifiers or surfactant materials. The soap reacts with lipid very fast. It is less than 1 minute. It will disintegrate the lipid, and immediately it will kill that virus or bacteria. This is a very simple mechanism, accepted mechanism across the globe. Now, my formulations, I have sent 4 formulations for testing in Indonesia, in your place. And my friend Dr. Yani is there. He'll be taking forward with these formulations. Here also I'm trying, and it has been sent to the authorities, concerned authorities there, and it is under trial. And once it is validated, the test will be validated, there will be no requirement for any type of drug regulations and all other clinical trials and all these things, because all formulations are made out of edible components. All are edible components. All are under FDA. All are under FDA, US list. So there will be, I will disclose everything after the NDA will be there, and I am telling it. I have, whatever I have done, initial trials over here, it is really encouraging, and I am very much confident about these products. it will show results on SARS-CoV-2 similar to the other viruses and bacteria, which are the lipid layer on the body. And as early as possible, I will request the authorities there in Indonesia, because whoever doing it, I don't have any object. There will be the chance that you can save the humanity from these dreadful disease. We cannot put out us under lockdown situation for years after years. It is not possible. Even for months in India, the lockdown is under lifting. They are trying to lift the lockdown due to the economy. The economy is also another major factor that has to be taken care of. So the people are getting infected very rapidly in our places. Now, let me coming to the mode of action. I have told you, that is in very brief, I have told you. I'm not going into elaborate descriptions of these things and all these things, make ingredients and all these things. As I have told, all the FDA approved materials I'm using, once it is validated from the virology laboratory on SARS-CoV-2, then it will be hours to bring it to the use of the people. The methodology of use will be oral and the nostril, the buccal cavity and the nostril cavity, from where mostly, and the eye also, a part of it, from where the viruses goes inside our body and infecting us, or the already infected persons, the air coming from their mouth, from their lungs, they are infecting the environment, they're infecting the uninfected persons. If they're putting these one or two products in their mouth or in the nostrils, it will kill the viruses while in entry, while it is entering into the mind. There is a study that it retains or it stays inside the nostril mucous membranes or the nostril cavity for a few hours. It stays there, then it goes when it gets chance to go inside it. And mouth cavity up to the tonsils, it stays there for a few hours. In accidental cases, not. In the general cases, I am telling. So I am claiming with full confidence, after a lot of inventions across the globe, many years, these formulations will save many, many lives and control the infection to a considerable percentage. So with your cooperation and under the situation of this pandemic, SARS-CoV-2, which is highly contagious and that it is killing very high population across the globe. So there is a pride, and I am telling that this is almost certain. These type of formulations, very easy formulations, and the ingredients are easy. And I propose and I request all of the authorities there who are listening over here, let us go ahead and have it tested on the SARS-CoV-2 directly and see the efficacy of this material, how it kills the SARS-CoV-2 there. And then we'll go within earliest period of time, not in years, not in months, but in days and hours, that to bring these formulations, after all formalities will be obviously completed, for the use of the people. This is my very small submission here. Thank you very much. I am ready here to just take your questions or anything else. Thank you very much, everybody.
Okay. Thank you, Professor Dr. Nilanjan Deb, for your presentation, for your brief presentation. I think we'll have another information from Muhammad Yani bin Absam PhD to complete the presentation from Dr. Nilanjan. I think now I would like to give a chance to Dr. Muhammad Yani Abu Sama, but previously I would like to show a little bit about his CV. This is it. This is the CV of Dr. Muhammad Yani bin Abu Samah. He's a Singaporean citizen. He has a lot of experiences as in industrial and in engineering. So he had this PhD in applied material science from Pennsylvania, USA. He's a master of engineering from about power system engineering from the same university. And before that, previously he has his MBA from international finance management from Sri Karian Wirat University from Thailand. Um, he lives in Singapore, but I think he has also a lot of experiences in any countries around this Asian, including Indonesia. But I think it's time to Dr. Muhammad Yani to present his papers. Dr. Muhammad Yani, now is your turn, please.
Okay. Sorry, I was unmuted just now. Okay. So let me continue.
Okay, go ahead.
In fact, I had been collaborating in several programs, including University of Medan area. We are, in fact, introducing a new method of dealing with environmental dilemmas. Okay, so basically, our proposal on this webinar event, which is basically touching on COVID-19 and also the mitigation programs that can be taken place, okay, to the immediate or to the more safe manner, especially when you see that what had happened to our world, okay, that comes back to the early part of this year, okay. So the thing is that myself and Dr. Nilanjan, okay, we have been dealing quite a lot, okay, in the development of several pathogenic killers, okay, namely because we are dealing with waste management and we deal in abundance of getting rid of pathogens, making our world much more safer to live on, okay. And the thing is that, okay, right now we are, especially nations, big nations like India, Indonesia, okay, and the Philippines, okay, as you can see what happened in America now, okay, all the governments, I can say all the governments in the world are facing the biggest issue, that is to implement the social distancing, okay. So we had come out, okay, with a kind of simple solution which can be applied, okay, to all the levels of societies, okay. Because right now, like Malaysia had just lifted the lockdown about a couple of days ago, whereby the social distancing, in fact, okay, is a big challenge for each and every government to implement. So and everybody knows that the COVID-19, okay, is a very, very contagious, very infectious kind of virus, okay, that can be transmitted easily through bio aerosol transmission, okay. So, and after the lift down has already been, okay, after the lockdown has already been lifted, but the next pandemic that we are going to face, okay, is the coming back to the original situation where the virus emerged, okay, last year's December. So what we are facing today now is basically the not social distancing, but it's the social pandemic that we are going to face in the coming days. Okay, it's not months or years. So Dr. Nilanjan and myself, because Dr. Nilanjan has a very good lab facility, and we see that the simplest form that the virus can be killed is basically utilizing the kind of soap or detergent, in order to puncture or break the lipid bilayer of the virus. Okay, so that is basically a very, very simple approach, that is to make soap and detergent edible. Okay, that is the main purpose of the development that we had already created. Okay, and the samples is already, is already with us, and we are for Malaysia, we are planning to give the sample to the Ministry of Health and also the Institute of Medical Research. Okay, whereby in India it has already been submitted to National Institute of Virology, okay, and also the ICMR, which is the Indian Council of Medical Research. Okay, the main purpose to this is very, very simple, because we want the remedy or the prophylactic, okay, remedy to be accessed by all levels of society. Okay, when you talk about advanced technologies like the development of drugs or vaccine, okay, to me, vaccine will take no less than 10 years to actually develop, because, vaccine, you have to send all over to the world and get it tested. Okay, whether it is, whether it is effective to a different kind of communities or race that we have in the world. Okay, so basically our remedy is very much straightforward. Okay, we just use the thesis or the concept of how soap kill the virus. Okay, but unfortunately the soap that you have in your bathroom, okay, the soap that you can find in Indomaret or Alphamart cannot be consumed. Okay, so we develop a system whereby, okay, the soap can be consumed safely, okay, to all levels of people, that whether they have, okay, some chronic diseases in their own, existing in their body or not. Okay, so, and because the material is not rocket science. Okay, rocket science means, okay, you need many years to actually develop this kind of system of curative approach. Okay, it can be immediately available, okay, upon acceptance from whatever authority is concerned. Okay, so, and I am very, very delighted, because I myself feel that this is a very big challenge that all the government, especially in India and also Indonesia, okay, to actually lift the lockdown in the most safe manner, because the hidden enemies or the commandos of the viruses are the people who are asymptomatic. Okay, these people are actually carrying the virus, but without us knowing who they are. So if the application of this curative approach, okay, is being taken, okay, by the person who is healthy, okay, if the person that is healthy takes the remedy, okay, it means it is creating an environment where the virus cannot make it as a habitat. Okay, but for the person who is already asymptomatic, okay, this virus, okay, this remedy actually kills the virus in their facial cavities. So whenever they talk, they cough, they breathe, they sneeze, okay, the bioaerosol transmission or the virus content and counts in the bioaerosol component will be very much reduced. Okay, so that is the main goal of it, because to us this is a national and international effort. So, and people like us, especially in the working societies, as you can see, the societies in Indonesia, it is almost impossible for us to actually implement the social distancing measures, because the culture is already like that. Okay, the same as India, when the government lift the lockdown, everybody start to run and sit on top of the bus and trains. Okay, and you see what happened in Brazil. Okay, the number of infection rises simply because of their ethical cultures. Okay, because they can't okay, they can't be a meter apart, because you know the way they embrace people, okay, they embrace their friends and families is through hugs and kisses. So these are the things that, in fact, okay, that actually prevails, okay, in our multicultural communities worldwide. Okay, so in order to prevent this, okay, in order to put a stop to it, okay, we should think of something which is can be conducted immediately, because you know social distancing, I think in the coming months or in the coming weeks is going to be a social pandemic. You know, I'm a Muslim and everybody thinks that somebody that probably has the virus will be a pig. Okay, oh, don't go near them because you know these people are virus carriers. So it will be better for us, okay, to put something which is a little bit immediate, okay, that can be you know, that can be straight away implemented. Okay, the samples are with me. I had already spoken to Pak Karim. Probably I will be sending some of the samples down to University Medan area probably next week. Okay, and I believe as a university that has a very strong biological department, okay, University Medan area can actually do the screen trials, okay, because you have people who are in the quarantine, and some people has already been infected by the virus there, okay, and probably put this stuff in their mouth or in their nose or in their ears and ask them to cough out. Okay, when they cough out, okay, you check, okay, the virus counts. Okay, if let's say before taking this application, the virus count is 10,000 per ppm. After applying this thing, okay, the virus goes down from 10,000 to 1,500. Means there is an improvement. Okay, it will be better for us to actually see what is our basic livelihood stand, because right now I think, okay, because everybody is afraid because of the infectiousness and also the quickness of getting infected by the COVID-19, and many countries in Southeast Asia has you know, is still closing their borders. Okay, remember, you know, we have sometimes we have families. I have also many promises to make to UMA, okay, because there are plenty of projects stranded there. So you know, this is one of the remedy that we bring forward. Okay, and I hope that we should be able to send okay, deliver the samples to University Medan area next week, and then let the university do the necessary testings, and if there is a positive effect from it, then we may think of putting this throughout Indonesia, Malaysia, Singapore. So let us concentrate on this region first, whereby we can open our borders and start to walk freely like before. Okay, so thank you very much, ladies and gentlemen, okay, professors and rectors, for giving us this opportunity. And I think you know, there are plenty of efforts in the world, you know, producing vaccines, you know, medicinal cure, but those are still on shelf. But what we have now is already in our pocket. Okay, so let us go for it, and thank you very much. So if you have any question or anything, please just ask. You know, myself and Dr. Nilanjan is more than ready to answer questions.
Sorry. Thank you very much, Dr. Moyani, for your presentation. Now, I think we have already done for the presentation session. Now we are going to Q&A session. As I told before that for this session, I would like to read the questions come from the participants, and I will ask the speakers to answer the question that related to his or her presentation today. I have already some questions, and I'll, for Prof. Diana and from Faisal Amri, because I think for Dr. Yani and Dr. Nilanjan, because just presented and then we don't have yet the question. But I think we will start the question to Prof. Dayana. Prof. Dayana, this is a question. I will read it for you. Prof. Dayana, I have a question for you. And are you still there? Prof. Dayana?
Yeah, Pak Zul.
Okay.
Yeah.
This is the question from Waluyo. Mechanism of action of coronavirus when infect humans, and how it can be asymptomatic, and does it mutate in human body? Have you already tested some natural compound that potentially can destruct the phylum of bacteria, and what the result is? This is the question. Can you catch it? Okay.
Maybe I answer the second question first. Okay. The second question is related with do I have already tested the compound? Yes, we did. So actually, we explore a lot of compound from bacteria, and this bacteria is isolated from marine environment and also from the phyllosphere. Phyllosphere is the surface of the leaf. So we see a lot of bacteria which have compound to inhibit or destruct the biofilm of bacteria, and we found a lot of compound that from this isolation for inhibition and also for destruct the biofilm formed by a lot of bacteria. And this bacteria we test first the one that opportunistic pathogen like Staphylococcus aureus and Pseudomonas aeruginosa, and also the one related with gastroenteritis like enterohemorrhagic E. coli, Salmonella, and the one with infection is Streptococcus pneumonia. So we found actually there is a specific correlation. For example, if one compound can only inhibit or destruct specific pathogenic bacteria, so there is a specific relation. At the time, we has toxicity assay using strain lethality assay. But this the next step to be done with clinical trial. But we want to use it as for environment. We already use it to apply this compound. But for human, of course, need to be more study. That's thank you, Basil. And the first question is. I'm sorry. Could you please repeat the first question?
Okay. The first question is mechanism of action of coronavirus when infect humans, and how it can be asymptomatic, and does it mutate in human body?
Okay, so how it can be asymptomatic?
Yeah, asymptomatic.
Actually, so far there are a lot of coronavirus strain. There are a lot of type of coronavirus strain because we know that this virus they have material genetic is RNA, so it is very easily to mutate it. So far there's a lot of strain of this coronavirus. So in our assumption, this also may be one of the reason for some people show, and this also one of the reason maybe for several people not symptomatic, and other with the condition is the disease is isolated. So firstly is the genetic property of the virus, but we also have to consider that the genetic property of the human as a host, the immunity as a host, and also we need also consider microbiota because it's also give stronger immunity for some people. So actually, to explain what happened. Thank you, Zulu.
I would like to read the question for Dr. Faisal Amri. The question is like this. Graphene can be used in combating with coronavirus. My question is, do this mean that graphene as a material can be used to detect coronavirus existence in our body? Or can it be used to protect us from coronavirus as a material to protect us from inhaling virus through our respiratory system, or it can be used as a material used to cure the disease? I don't know what does it mean. I think Dr. Faisal can catch the idea. Is it possible to use graphene for HIV treatment? So this is very different. And does it have negative effects on human body? So this is the question, and I think now, please, Dr. Faisal Amri, answer the question.
Thank you, Prof. Zul, for the question. I think all the questions go to one summary. Actually, is it graphene applicable to be used as the sensing device to combating COVID-19 virus? I think like we have discussed earlier in the presentation, that some experiment has been proven that this kind of material can be used as the material to detect or to recognize particular virus when it comes in contact with the surface of the graphene. So that's why one strategy to enhance the recognition ability of this material is through the biofunctionalization with the similar molecule that can be detect the virus itself. So the surface of the graphene is not only as the surface to detect, but also as the biomarker and the biomimicking surface that can selectively detect a particular biomolecule. So it's expected that this material can be used as the rapid test for the COVID-19 virus, replacing our current PCR, because PCR I think it take around 1 to 2 weeks for the result come out. But using this kind of material, it can be used a rapid test where the result can be obtained immediately. Okay. But the problem is when we consider to use this kind of this material to combating COVID-19, there's that the problem is rise from its toxicity level, because we know that this material coming from carbon, and we know that the carbon is have a certain level of toxicity that we must be further study. Is it this material edible or not? That's the main problem now, because the toxicity level is still as a matter of debate now by the many researchers who study about this kind of material. But for the in vitro analysis, it's a good material actually, and low cost material as a rapid recognition for the COVID-19 viruses. That's my answer actually. And then can this material be used to combating HIV virus? Actually, basically, whatever of virus can be detected by using this material. But to study more about this ability to break down or to rupture the surface of this membrane, is also can be used. But the problem is it cannot be used as for the oral intake application, like ordinary like a hair for example, like a conventional drugs that we can intake. So that's the problem. The toxicity level is still a matter of debate. And secondly is about what is the yeah, since this cannot be used in vivo experiment, the most of the application is as to detect or as a sensing devices. So that's why many of the research now they aim that we use to mitigate the spread of this virus from the outside, like the use of this material as the face mask material that can absorb the virus bioaerosol, and then it can rupture the virus, so it can it's no longer dangerous to human body anymore. So that's my answer, Prof. Joe. Thank you for the question.
Sorry. Thank you, Dr. Faisal. Now I'd like to address one more question for Dr. Nilanjan. This is the question. I'll read it for you, sir. For your presentation, we gather that you have already developed a natural cure for the destruction of the SARS and CoV-2 virus within the human body. So this will be safe for ingestion as it is already made up of edible substance. How fast and how easy will it be to manufacture the cure for mass production once it has been sufficiently tested, and how soon can we realistically see this in our markets? Can you catch it, sir?
Yeah, very.
Okay.
I understand the question, and the question is very clear.
Yeah.
And I am, in a very brief note earlier, I have given the concept of the cure or the medicine for that. Yes, the time required for these to be reach the patient or the person or the people who will willing to use these things will be hours to days. I have mentioned it earlier also. So it is so fast that we can market it as far as we can produce it after the validation that it has genuinely worked on SARS-CoV-2. I am just absolutely targeting SARS-CoV-2, COVID-19. So all other viruses and bacteria, they are in the line. Now I am targeting SARS-CoV-2. So as you have asked the question, that these are the substances which are edible, and they are the formulations, ingredients are there which are easily available across the globe, and the manufacturers can manufacture the material as per my guidelines and my formulations. And after we can get, first I will propose that whatever the formulations I'm suggesting, we will manufacture to the best that I will be directly involved into that manufacturing, and then we will provide it with that sufficient protection to the people for use. And that another thing I am adding, which is not there in the question, that how long it will work when you are putting inside your buccal cavity. I have tested, so for my test, it is minimum 2 hours. A single dose will protect your mouth cavity, and single use in the nostrils, both of the nostrils will protect you 2 to 4 hours inside your nostrils from getting infected. So I think that this is the answer so far from my side.
Okay. Thank you, Dr. Nilanjan. I think all the questions have already been answered by the speakers. There is no more question here. I think we are about to end the webinar. Previously, I would like to express a little bit pointers that I can get from the presenters today. I don't mean it to cover all the content of the presentation, but I just remind us that what are the points of each of the speakers that present today. From Dr. Diana, I got 4 points. Secondary infection are more likely to happen, especially for hospitalized patients. Bacteria could naturally from biofilm. Thus, the bacteria will be coated by biofilm, which make it resistant to antibiotic. Anti-biofilm is needed in the therapy to overcome the resistance problems. Anti-biofilm also could be extracted from plants. So from Dr. Faisal Amri, I got 2 points. Graphene can be used to enhance the efficacy of drug that used to treat coronavirus infection. Graphene oxide derivatives reported to be effective to fight strain of virus. And from Dr. Yani and Prof. Nilanjan, there's I think, interesting thing. Edible soap is a new method to fight coronavirus, which will be useful in upcoming new normal phase. But right now, don't try to eat soap. Don't try to eat regular soap. So I think that is the summary of our presentation, to our seminar today. Finally, we have reached the end of this event. Once again, I would like to thank to all the speakers, Dr. Nilanjan, Dr. Yani, Prof. Diana, and Dr. Faisal Amri. Thanks to all the participants also and all parties that involved for this webinar. If there is something wrong, this comes from me, and please forgive me. Hopefully, the webinar will benefit us and the community so that the spread of COVID-19 can be blocked or even can be stopped totally. Wabillahi taufiq wal hidayah. Assalamualaikum warahmatullahi wabarakatuh. Good afternoon. Okay, done.
Medan is the capital of North Sumatra province. Medan is the largest city on Sumatra Island with the 3rd largest population in Indonesia, which consists of various cultures, ethnic groups, religions, and languages living in harmony and humanist. Medan is a city with a strategic position adjacent and surrounded by